Hello, hi. We are doing the survey of fungi module. We have already completed discussing certain topics in fungi like the life cycles in Zygomycota, that is Rhizopus and Mucor. Then we started with Ascomycota and we have to see these many life cycles. 1, 2, 3, 4, 5, 6 life cycles, out of which Saccharomyces we have completed in entirety. Then Aspergillus, we have already completed the general account, thallus structure, and asexual and vegetative reproduction in the previous lecture. Today, we are finalizing or finishing Aspergillus by completing its sexual reproduction and economic importance as well. Then we will be seeing Penicillium, Neurospora, Precisa, Claviceps in the upcoming sessions. Then after that, we will be seeing the life cycles in Basidiomycota, that is Puccinia, Agaricus, and Rustilago. Then we will be moving to fungi-like organisms, that is Mycota, Phytophthora, and Albigo life cycle we have to see. Then we will be seeing lichens and mycorrhiza, fungal toxins, etc. So our today's agenda is this: sexual reproduction in Aspergillus fungi. Okay, we have already seen the sexual reproduction part, and today we are going to see the sexual reproduction. Okay, how it proceeds and all. So before we begin discussing sexual reproduction, this is just a few things about Aspergillus. Okay, it's a common fungus. It's almost ubiquitous. You can see it everywhere in soil, water, food material, air, everywhere you can see it. Around 100 to 500 species are not. Some species are economically important. Some species are pathogenic. So general features of Aspergillus. Okay, it's black or green mold, and since they are producing some sacs for carrying their spores, it is called sac fungi. That's why it is brought under Ascomycota. Okay, then mode of nutrition is heterotrophic because chlorophyll is absent, so they will be doing like saprophytic mode of life cycle. Then their reproduction is through two types of spores. In a sexual reproduction, we have seen conidiospores, and after sexual reproduction, it will be producing ascospores. Then it is actually a terricola species. Mostly it is found in ground or soil. So it is saprophytic and parasitic. Then like normally what happens, like when the vegetative mycelia absorbs nutrition from the environment through the vegetative hyphae, then it will be growing to produce reproductive structures called spores, like ascospores. We will see how the vegetative mycelia is going to produce ascospores. That is the agenda of today's discussion. So there are two types of hyphae, vegetative hyphae and reproductive hyphae, and the reproductive hyphae will be producing the fungal spores. So let us jump into today's sexual reproduction discussion. There are different species of Aspergillus which is showing diversity of sexual reproduction process, and it is completely and perfectly studied in Aspergillus herbaceorum. Okay, Aspergillus herbaceorum. Then during sexual reproduction, what happens? Sexual reproduction, there will be, this is the normal vegetative mycelia. During the sexual reproduction, there will be the formation or development of male branch, female branch, and male branch. Okay, male branch and female branch will be developed from the normal vegetative mycelia, and the female branch is called archicarp and male branch is called polynodium. Okay, and what will happen is that this, like after this particular female and male branches cut off from the vegetative mycelia, it was initially actually unicellular. Okay, multinucleate. Okay, all the entire cell is multinucleate, so it is multinucleate, but it was aseptate. So unicellular, it was also aseptate. But suddenly what will happen as the time progresses, there will be development of two septa and three cells in the body of archicarp. There will be a top elongated cell which will be the receptive filament for absorbing or receiving the male nuclei, that is called trichogyne. Then there will be a middle elongated cell that will be called ascogonium. Then there will be a stalk cell. Now comparing that with the male branch, the polynodial branch, there will be only one septa, so two cells. The top cell will be acting as the antheridium, which is also multinucleate, and there will be a bottom stalk cell. As the time progresses, this archicarp tightly coils, like spiral. It's like a spring. Okay, it's like a screw otherwise. Okay, completely coiled, and only the top area will be visible. The trichogyne will be visible, to which the antheridial branch will come and attach. So there will be the connection between antheridial tip and the trichogyne, and the entire content of the antheridium, the entire multinuclei and the cytoplasm of the antheridium will be transferred to the ascogonium through the trichogyne. One thing you have to focus here is that after this structure is formed, okay, this antheridium and trichogyne has fused. Now this is a like a reproductive structure like this, right? Now it will be completely covered by sterile hypha, which are arising from the vegetative mycelium that is lying beneath. Okay, you can see that this dotted line. So sterile hypha will be continuously covering it. Since it's a reproductive structure, we have to protect its male and female nuclei, right? So it will be completely covered by sterile hypha rising from the vegetative mycelium. So this forms an encircled structure. Okay, this structure will be having an outer layer called the peridium and an inner layer called the tapetum. Tapetum will be providing the nutrition. Now you can see that after the male nuclei content is formed, is transferred to the ascogonium, you can see there will be pairing of male and female nuclei. Okay, initially there won't be any fusion. I'll tell you when it will be fusing. It will be fusing it very late. But before that, after the male nuclei and female nuclei, both ascogonium is multinucleate, both antheridium is also multinucleate. So there will be many female nuclei and many male nuclei within this particular body. I'll show you here. Okay, you can see that. So it got transferred. So there will be pairing up of male and female nuclei. Can you see this? Focus only on where I am pointing. There will be pairing up of male and female nuclei. Initially, it was not segmented like this. It was also completely unicellular and sinusoidal. But after the pairing up of male and female nuclei, there will be segmentation or cell formation. You can see cell formation. So this particular hypha, which is having segments in which within each segment male and female nuclei paired up, this is known as ascogenous hypha. So from where did it arise? You can see that there was an antheridial branch, there was an ascogonial branch. So the tip of antheridial branch and ascogonial branch fused, and there was plasmogamy. This is the plasmogamy. Okay. Now from the plasmogamy, plasmogamy means what? Just pairing up of male and female nuclei. From such branches, from that body, from the ascogonial body in which all cells are paired up, there will be the formation of segmented ascogenous hypha. Okay, it is segmented, right? Segments, one segment, two segment, multicellular type. Okay, so now what will happen is very, very interesting and very important, and this can be a separate question as well. At the tip of each ascogenous hypha, something is going to happen. So at the tip of each ascogenous hypha, the thing that will finally happen is like this: zygote formation. Both nuclei at the tip, only at the tip, the nuclei will be fusing. Then there will be meiosis and there will be formation of ascus, and within that you can get ascospores. But before the formation of zygote, just focus at the tip of this ascogenous hypha. What will happen is that, imagine this ascogenous hypha, this tip topmost cell is this one. Okay, this is the ascogenous hypha. So from before reaching the zygote, from this cell come to here. Okay, this is the tip of the ascogenous hypha. It will be at maturity during sexual reproduction. It will be forming a hook. This hook is known as crozier. So one cell will be moving within the hook, and the second cell will be staying behind. And now each of the cell will be dividing. Okay, the red became 2 and the green became 2. Now there will be a septa formation. Okay, now there is a hook cell and there is a normal hyphal cell, and there is a penultimate cell. This is known as penultimate cell. This is the ultimate cell. This is the penultimate cell. At this penultimate cell, something will happen. In this penultimate cell, from now it will be called as ascus mother cell, because ascus will be formed from this particular cell's development. So this penultimate cell now fuses. Okay, now it fused. You can see it fused. Now you can come to this point. Okay, after fusion, it is formed a diploid zygote, and this zygote will be this zygote containing sac. Okay, this zygote containing penultimate or ascus mother cell, that will be forming a sac-like structure. I'll show you this in the previous slide. Okay, this ascus mother cell now formed a sac-like cell structure. Okay, it is going to be converted into an ascus. Okay, you can see the diploid nuclei or zygote within this. Now this zygote undergoes what? Meiosis. So after meiosis, there will be 4 ascospores formed. Okay, actually 4 nuclei formed. Each nuclei will be accumulating cytoplasm and forming a wall around itself. So it leads to formation of ascospore. Okay, after meiosis, it will be forming mitosis. Then only it will be forming by absorbing protoplasm and cell wall, it will be forming an ascospore. So 8 ascospores are formed. So you just imagine, it is only from one particular ascogenous hyphae, from one particular cell of the ascogenous hyphae, one ascospore formed. I'll show you in the other figure as well. From one, this is one such cell, this is another such apical cell, this is another apical cell, this is another apical cell. So from one particular, from each cell of the ascogenous hypha, one particular ascus will be formed. Okay, so such many ascus will be covered by the peridium, and you can see this is one ascus, this is one ascus. Every ascus are having this particular structure called ascus. Okay, this is ascus, this is ascus, this is ascus. So these many ascus are formed. Okay, we are not showing that ascogenous hypha here, we are just showing the ascus structure. Ascogenous hypha is behind this thing, but we are not showing that. So finally you can see that ascogenous hypha will be separated from the ascus, and you can see the male and female nuclei within the ascus. Okay, this male and female nuclei, that will be converted, that from now it is converted into ascospore. Now it is the male and female nuclei that you are seeing will be accumulating protoplasm, and they will be getting a cell wall around themselves. So that will be forming ascospores. Within each ascus there will be 8 ascospores, and each ascospore is a pulley shaped structure. Okay, you might have seen pulley from which we will be using to lift water from the well and all. It's a pulley type shaped structure, and each ascospore will be germinating at the time of maturity. Okay, it will be this particular ascocarp wall will be dehiscing, and ascospores will be released out. But before that, before reaching this stage, let me tell you two more things. This particular peridium covered structure, which is having many ascus, this structure is called the ascocarp of this particular Aspergillus fungi, and this particular ascocarp is a closed body with no opening. You can see this. Okay, this structure is known as cleistothecium. So cleistothecium is the fruiting body or ascocarp of Aspergillus fungi, and within that we will be getting many ascus. Each ascus will be having 8 ascospores that will be at the time of germination, that will be released outside, and it will be forming a new germ tube, and this germ tube will be developing itself into new mycelia. Okay, new mycelia, just like vegetative mycelia, which will be again forming the male and female branches, and that will be ultimately forming the cleistothecium, and the cycle repeats. So I have attached everything in this. Okay, this is the after meiosis, this is after mitosis. So this is the ascus. This ascus will be it's not an elongated body like this, it's a spherical body. You can see this we are showing it as a spherical body. It's actually an elongated body, but we are depicting it in this way for visual clarification. We are showing it like a spherical body. This is the this is the ascus. So that will be germinating, and that will be releasing the spores and all. So I have, you can pause the video. I have given sentence by sentence explanation in each slide. So you can pause the video and take the notes. I have already explained all the slides. Okay, just pause the video. This is another image given in textbooks and all. So I have already explained what happens during fertilization and all, and this is the development of ascocarp, crozier formation, everything explained. Every single thing I have explained. Just pause the video and take the note. And one thing you have to see, one note. Okay, this note I will explain in the upcoming slide. So there is a different formation of sexual reproduction in different ascomyces species. In some species, the antheridium is completely functional, and it is observed in Aspergillus herbariorum, and that will be doing the fusion and fertilization. But in some species like Aspergillus repens, antheridium is developed, but its contents are not transferred to female. So only the female nuclei will be pairing up and forming dikaryon. In some other species, male nuclei will be degenerating by the time antheridium has reached maturity. So such things also can be observed. Now these are the economic importance. You can pause the video and take the note. This is the harmful aspect. So with this we are winding up. Thank you.